In the second section of Chapter 4, Genes and Genomes, we're going to talk about different types of non-coding sequences. In particular, we're going to discuss two different types of RNA, one known as microRNA and also known as link or long non-coding RNA. We're going to talk about different types of repetitive DNA sequences, which are going to include what's known as short intersegmental elements and long intersegmental elements, also known as signs and lines, respectively. We'll talk about different types of transposable genes. Certain types of genes that are going to be moved from different sections of the genome that can either affect a heritable disease or different types of cancers that can pass down from generation to generation, or can help express certain phenotypes that actually may be beneficial to the individual organism. Finally, we'll talk about different types of gene duplications and what's known as pseudogenes. When different types of genes get transposed on different areas, oftentimes they're not coded with the same start sequence or introns, and oftentimes they become latent or unusable and become what's known as pseudogenes. Very similar to different types of retroviruses, we'll be taking a look at how this mechanism actually unfolds along the genome. First, we'll look at what's known as microRNAs or miRNAs, which are about 22 nucleotides in length. Once these are transcribed, these will actually form into a hairpin configuration, and they will be cleaved by two different nucleases, drosha and dicer, and it's going to fold itself into a particular type of mRNA target known as the RISC, or the RNA-induced silencing complex. So very simply, the job for these particular microRNAs is to help inhibit translation and stimulate different types of degradation of mRNA. So if we have an mRNA that is simply starting to fall apart, where the amino acid chain is starting to degrade, then very simply the miRNA will help degrade and block any further transcription of this particular mRNA sequence. So here we see a close-up look of the DNA for the microRNA gene transcribed into the hairpin loop that you see here. So this is a pre-microRNA. It's going to be cleaved by both the nucleases drosha and dicer to form the microRNA duplex as seen on the bottom here. Now, as you can see, this is atypical of what we usually think of RNA, especially messenger RNA, as a single strand of nucleotides. In this case, the microRNA is formed as a duplex. And of course, it will seek out the RISC complex, otherwise known as the RNA-induced silencing complex. So this, in effect, will block any particular mRNA that simply is not effective or possibly mutated, to prevent or at least suppress any type of translation, and basically to stimulate the breakdown of this particular mRNA molecule. Now, let me explain exactly what ENCODE is. Very simply, ENCODE was a project that was developed back in the mid-2000s. Back in 2004, the term junk DNA was thrown around. Once we figured out the length of the human genome, and knowing that we only had about 20,000 or just over 20,000 protein-coding genes, we figured it was such a small part of the genome that the rest must be junk DNA, or therefore non-usable DNA. Well, back in 2007, there was a number of different scientists, about 400 in fact, over about 30 different laboratories in the United States, that conducted experiments to map out the genome and basically look at any metabolically active sites found on the DNA. And what they found was kind of surprising. What they noticed was just under 75% of the DNA strand is going to code for a combination of protein-coded genes, which we know of already, amino acid chains to form mRNA. But what we didn't realize was there are other parts of the RNA sequences that will bind for either what's known as the short non-coding RNAs or long non-coding RNAs, or what's known as pseudogenes. So these will encompass the transfer RNA, the ribosomal RNA, but also the different types of RNA we talked before, including the link as well as the microRNA. So what they found was that there is more of DNA that's metabolically active for transcription, but it's simply not just for protein-coded genes. So through further research, and this is still ongoing, so they're still trying to hammer down the actual percentages, as it has been changing over the last couple of years. But very simply, the function for the other non-coding RNAs and also the pseudogenes, which we'll talk about in a little bit, have a great influence on gene regulation. What they also surmised too was, due to the complexity of the human organism, it's needed that particular type of gene regulation to turn certain things off and certain things on again as this particular organism develops. So as we know, if you know anything about embryology, of course, certain types of sequences have to be turned off, basically to have a male turn into the male part of the species, whereas a female, they let certain things go. So there needs to be certain types of regulations on these particular types of genes to be expressed. So what we thought was before, the rest of the DNA, roughly about 8.1%, is going to be the genome binding transcription factors right off the DNA. So those are the proteins that regulate transcription itself, not necessarily translation. But the scientists even admitted too that the 8.1% is kind of low. They do expect that number to go up considerably regarding the genome binding transcription factor. So again, this ENCODE testing is ongoing. I'm sure those percentages will start to even out a little more so as the years go on. While microRNA will inhibit and also degrade mRNA, if you look at link RNAs, known as long non-coding RNAs, these will actually inactivate almost the entire chromosome. In this case, we're looking at the X chromosome being inactivated by what's known as link RNA ZIST. So very simply, this is going to be around 200 nucleotides long, a lot longer than what we saw with the microRNA. So this will help regulate by inhibiting expression on a particular enzyme. In this case, it's going to inhibit the second X chromosome that you'll find in female cells, which is simply, it's redundant and simply not needed since there already is an X chromosome is being used in the female organism. So very simply, it locks this X chromosome in place, which we have seen if this link RNA exists, it does not exist, then we see certain types of disease processes occur in the female end of the species where both X chromosomes are wildly expressed, which can lead to different types of, if both X chromosomes are active, this can basically cause an inherently fatal condition as there'll be double the amount of proteins that will be activated, which actually will throw off the different hormones, throw off different regulations of those hormones and development of any kind of primary and secondary characteristics. Now, this is only one example of the link RNAs, but from what research has shown us, there's a far greater number of link RNAs than are actual protein-coded genes. So very simply, this suggests that the non-coding RNAs make a substantial contribution to the biological complexity of the higher eukaryotes. So again, just having a certain sequence of certain genes being expressed to allow proper growth or to allow differentiation for different types of cells all throughout the body is inherently active or very simply controlled by these link RNAs. So here we see a direct comparison of the amount of protein-coded genes we have on the genome compared to the link RNA genes as well as the link RNA transcripts. So looking at familiar species we looked at over the last couple of chapters, starting with yeast. So you see the base at a 12 megabases along with the genome size, about 6,500 protein-coded genes, with about 53 link RNA genes and 55 link RNA transcripts. So you see a significant jump to the worm C. elegans, then also to Arabidopsis thaliana, which has the largest number of protein-coded genes, but not quite as many link RNA genes we get to the higher organisms here. And as you see, not much more on the link RNA transcripts. Now, when we get into the animal kingdom, especially with Drosophila, just simple fruit fly, slightly more of a gene size, but you see less protein-coded genes, but you see far more link RNA genes. So very, very important for the development of the Drosophila fly. As you can see, the link RNA transcripts are almost 3 times as more than the link RNA genes. So again, the link RNA transcripts are tightly controlled. So the link RNA genes are tightly controlled to allow the protein-coded genes to be effective when they need to be. And of course, as you can see, when we get from the mouse to the human, it's quite a significant jump. When you look at us in particular, genome size of 3,100 megabases with only 20,000 protein-coded genes, almost 5 times more link RNA genes, and you're looking almost at 9 times more, probably a little bit less, link RNA transcripts. So when you think about the development of the baby, once you start at fertilization, how many things have to go correctly at the right time for the right sequence in order for the baby to grow inside the womb and actually be born, it's really intriguing to look at how this whole thing is totally controlled. And this gives us relatively a good idea of how complex the human organism is and why we have such a tightly controlled system in place to make sure that particular organism is survivable. Now, differentiating from the microRNA and the lincRNA, we have what's known as repetitive sequences. So these are usually long repeats of adenosine and thymine bases, which can take about 10% of the sequences found in humans. Now, conversely, these are not going to be transcribable areas of DNA. These tend to have more of an influence on how the actual banded structure of the chromosomes come into play, how it wraps around histones, and so on and so forth, which we'll get into at the last section of this particular chapter. So one particular type we find in the human body is known as alpha satellite DNA. This tends to be, again, very rich in the adenosine and thymine bases. It's about 171 base pairs, but again, only takes up about 10% of the human genome. The other types of non-coding parts of the genome that you see here include the retrotransposons and the DNA transposons. So the retrotransposons, again, are going to be repeated elements found within DNA. You have what's known as long interspersed elements known as lines, short interspersed elements known as signs, and also different types of retrovirus-like elements. So as you can tell, these behave very similarly to different types of retroviruses, and the most popular one that most people know about is the HIV retrovirus. Now, the main difference between the three retrotransposons will be size. So the long interspersed elements are going to be about 4,000 to 6,000 kilobases long, whereas the signs or the short interspersed elements would be 100 to 300 base pairs long. And the retrovirus-like elements tend to be relatively short, about 2 to 10 kilobases in length. So very simply, all three of them will insert themselves into the DNA by the reverse transcriptase, very similar to, again, the retrovirus that we see with HIV. And conversely, the DNA transposons, rather than being inserted in DNA by reverse transcriptase, tend to be copied and reinserted as DNA sequences instead of being inserted by reverse transcription. But as you'll see in the next few slides, that each one of these elements can have some kind of effect, cause any kind of mutation in the human genome, often with the different types of effects that can happen with the genes or the expression of those genes within the DNA. Oftentimes, this can be very detrimental to the organism, or in some rare cases, can be also beneficial. Now, this figure shows a representation of the signs, the lines, and also the retrovirus-like elements. Very simply, you see the retrotransposon is in the chromosomal DNA, it's transcribed, then through reverse transcription is going to form another section of the retrotransposon DNA, and then will be inserted on a different portion of the chromosome. In this particular site, is going to be different than the original chromosomal DNA, whereas the DNA transposons will actually be interspersed through different areas, will be copied and pasted, again, usually in a new area than the original chromosomal site. What we've learned about retrotransposons have come from our study of retroviruses. The main difference being that the retrovirus naturally is going to package its particular RNA sequence to be reverse transcribed into the DNA complex and integrated by what's known as integrase by the viral enzyme to basically perpetuate any further production of that particular retrovirus. However, we don't have that particular type of coding or any type of packaging that we normally see with the different types of retrovirus-like elements. So that's why we mentioned it's not quite a retrovirus itself, but it's retrovirus-like. So very simply, they don't spread to one another, but every time the DNA will actually replicate, it will pass down from one generation or the progeny down to the next. So very simply, the other lines and signs, the main difference being that the retro-like elements as well as the lines will use reverse transcriptase, whereas the signs, or the smaller integrated elements, very simply will actually be inserted by different types of lines. So again, the retrovirus-like elements as well as the lines will use different types of reverse transcriptase to be bound into the DNA, whereas the signs a lot of times will actually be integrated by either other types of RNA or oftentimes by lines. Now, all these transposal elements can have negative effect with gene expression and also be passed down to different generations. So very simply, the signs and lines can very simply disrupt the genes themselves or disrupt how the regulatory proteins are going to affect those particular genes. So certain types of diseases, which include hemophilia, cystic fibrosis, muscular dystrophy, as well as different types of cancers, can be augmented depending on where these transposable elements are going to insert themselves into the DNA. Now, conversely, sometimes these particular mutations can work to benefit the organism. And everyone remembers the moth study during the Industrial Revolution, where very simply the different types of transposed elements will often either inhibit or excite certain types of gene expression. So in this case, during the Industrial Revolution, we know about the light-bodied moths. When they land on the different types of soot-covered trees and other structures, they were selected by the environment to be hunted and found and eaten, of course. So therefore, they can't pass their phenotypes down to the next generation. Whereas the dark-bodied moths, which had a different coloration, which were coded for these different types of mutations from the transposable elements, were able to pass on their particular genotype and phenotype down to the next generation. So this is one of the rare cases where the transposable elements works to the organism's benefit, this particular type of organism within its population, to pass it down to the next generation. Now, besides the insertions of the retrotransposons, we also see different types of recombination in between repetitive sequences, where oftentimes we see different lines will actually cleave and then actually transpose themselves on different types of genes. So this allows for recombination between the lines, which again can either inhibit or possibly augment the gene expression of a certain type of gene. But this is responsible for the genetic diversity that we see throughout the different species, and also helps reshape the different genomes, helps us express different types of phenotypes, which again can either be naturally selected for the organism's benefit, or it could also be selected for the organism's downfall. In the final section, we will talk about gene duplication and pseudogenes. So gene duplication simply is that, multiple copies of the same gene over and over again along the same section of the particular genome. So oftentimes what we see in different types of organisms is each gene has a certain either target tissue or a certain target in development that does the same basic function. So here we look at the globin gene. So we're looking at the alpha globin and also the beta globin loci. So very simply, these are going to be clustered around the same region in DNA. And whereas some will be used more so in more fetal tissue, whereas some will be used more in adult tissue or different types of tissue as this particular organism develops. In this case, the embryonic tissue tends to have a higher affinity to oxygen than most of the adult globins. So very simply, it allows the embryo to derive as much oxygen as possible through the umbilical cord, through maternal selection. And notice too on this particular slide here that there are different areas of pseudogenes. So pseudogenes are going to be non-functional gene copies of the other genes that we see here. So these just simply take up space within the human genome. So usually they can be arisen by different types of mechanisms, such as one could be duplication of that particular segment along the DNA, or oftentimes the entire genome can be duplicated, as we see in the Arabidopsis genome. That's why we see the Arabidopsis genome being so significantly large compared to our own. Now, the second way you can achieve a pseudogene is through retrotransposition, where very simply there's a reverse transcription of mRNA, and this can be integrated into the DNA sequence. So very simple, what happens is this will often be inserted without the particular intron, which basically will help start the process of transcribing that particular DNA sequence. So this duplication can often yield inactive gene copy, but still be processed, known as a processed pseudogene. So this can also be an inactive gene as well. So the majority of pseudogenes that we identified in the human genome are known as processed pseudogenes. So you see here off the gene, you have the introns already being spliced out. So you're left with an mRNA that normally should be translated by a ribosome, but instead this goes through reverse transcription and it's integrated into a new chromosome site. Now, as I mentioned before, the introns have been taken out, so this will not code properly with the processed pseudogene, so you'll not be able to get a functioning mRNA or amino acid chain thereafter. Sometimes these pseudogenes can be actually expressed by what's known as a retrogene. So an example here, first without looking at dogs, if you look at the FGF4 gene, so you have the transcription and splicing, so you have your mRNA, and it's going to be first transcribed back into the DNA strand of the FGF4 retrogene. So there's your normal FGF4 retrogene now that we see in short-legged dogs. So in this case, this actually shuts down the bone growth to produce the short-legged dogs like you see here. So you've got different types of Basset Hounds, Dachshunds, and of course the Corgi to the left. That this particular type of gene actually just creates some new breeds for this particular line of dogs. So occasionally this can be a benefit just to add some diversity into the species, but this is rare compared to the other types of pseudogenes that we see. So again, this process pseudogene, same mechanism, but because it is actually somewhat functional, is known as a retrogene. So again, you can see again that from the FGF4 gene, you have normal mRNA is going to be transcribed and then, of course, reverse transcribed back into a different portion of the FGF4 retrogene without the introns. And of course, this will cause an abnormal expression of the leg length gene, the FGF4. So this wraps up the second section. We'll look to the next section, which will finish out chapter 4. Once we finish chapter 4, we'll be ready for our exam the following week.